Hello and welcome back to Anatomy and Physiology here with your professor, Dr. Shawn Clavell. And today, as relates to anatomy and physiology concepts, we're going to be talking about the skeletal muscle contraction. Particularly, we're going to talk about the major steps of the skeletal muscle contraction and the first step, which is excitation here. But let's kind of just review first the gross anatomy of skeletal muscles first. And we take a look here at the gross and microscopic anatomy of muscles. And so remember that muscles are just bundles of fibers, right? A whole skeletal muscle like your bicep, your tricep is made up of a bunch of muscle fibers that are bundled together in groups called fascicles. Let me see if I can't zoom in there and demonstrate that fascicle appearance there that we see. And it's a connective tissue covering, holds a group of muscle fibers together. But within each muscle fiber, what we're going to see is that there is a number of long protein strands called myofibrils. So the myofibrils are long strands of protein, and they contain two structures called the myofilaments. The myofilaments are actin and myosin. The thick filament is myosin, the thin filament is actin. So if we zoom into a muscle fiber itself, it has its own cell membrane. It's called the sarcolemma. It's got its own cytoplasm that's called the sarcoplasm. And ultimately, it's made up of these long strands of proteins, which are made up of these repeating patterns that we call the sarcomere. So, the sarcomere is kind of the basic functional unit of a muscle contraction. So, here's the sarcomere. Draw that right there for you. There's the sarcomere. And what the sarcomere is it's bound by two really strong proteins. These titin proteins create what's known as the Z disc. So this is just the end of the sarcomere, and a sarcomere is just a long repeating pattern of these. A muscle fiber is just a long repeating pattern of these sarcomeres. And so there's the Z disc there, and it anchors these two alternating bands. The thick filament is in the middle here. That's the myosin. Let's make it I don't know, let's make it orange. Okay, the thick filament here in the middle is myosin. The thin filament in the outer portion here, I'll make it green just as it is in the slide here, is actin. That's the thin filament. So you can see that there's this kind of alternating pattern here of thick and thin filaments. This is what makes up the ultimately the microscopic structure of a muscle. So if we were to even zoom into a single sarcomere, what we'd see is what you have here. You've got the sarcomere at the top of the screen. You've got alternating thin, thick filament, thin, thick, thick filament here, myosin in the middle, and the M line is really just the middle line. So when we look at this here, the M line identifies the M line. The I band is the region of the muscle where there is no overlapping actin or myosin. The A band is where we see all of the myosin. But what we'll focus on in this slide is the structure of both myosin and actin. You can see that myosin has a club-like head from its extending from its base. And so this is the thick filament, myosin. It's got a long club-like head, and and what you will see later is that it actually pulls that. I'll go back to the slide here. It'll actually pull the thin filament to the midline using its club-like head. Then we have the thin filament actin. Now, attached to actin are two regulatory proteins. Those regulatory proteins are tropomyosin. Now, tropomyosin is a long strand of protein that runs all the way across the actin and actually covers the binding site. So it covers the binding site for myosin. So when the binding site for actin, which is on myosin, is always going to be open because it's located on that club-like head. The binding site for myosin, located on actin, is hidden by a long strand of proteins named the tropomyosin. Then you have troponin. Troponin is a tri-protein complex that's responsible for maintaining the position of tropomyosin. If calcium, we've heard of calcium here in the skeletal muscle structure, it's located within the sarcoplasmic reticulum. But if calcium is released and binds to troponin, it'll cause a shape change, allowing for tropomyosin to uh, reveal the binding sites of myosin, and then we can start through a muscle contraction. So just as we start to back up here in terms of these slides, let's take a look at the thin filament actin. It's covered by two regulatory proteins. One is tropomyosin. It's the long protein structure that is responsible for covering the binding sites of myosin. The other one is troponin. That's the one that holds structure and holds shape. So when calcium binds to troponin, it allows tropomyosin to slide off and the contraction to occur. Myosin is known as the thick filament. It sits in the middle and has a club-like head structure. On the club-like head structure, you've got a actin binding site. So the actin and myosin both have binding sites for each other. And then there's also a place for ATP to bind. You can see this kind of twisted structure as well as the hinge region, which allows for a good kind of a muscle contraction. These myosin heads actually are pictured or mechanically pictured like springs. So when a muscle is contracting, it's almost like it's being wound up and then whipped forward in terms of that mechanical structure. And so the thick and thin filaments are in alternating pattern, with the myosin thick filament being anchored into the middle by the strong protein titin as well. The thin filament is anchored on the periphery here by the Z line. And so what we have is a sarcomere. That is the functional unit of a muscle, the smallest functional unit of a muscle. Ultimately, we can see that alternating thick and thin bands right into the muscle fiber here uh, in the sarcomere structure. And so a muscle fiber is a number of alternating bands that we see this sarcomere structure. And so we've kind of looked at the gross anatomy of the muscle here, and then we'll review the major steps of a muscle contraction. So, the major steps of a muscle contraction are excitation. So, that means that a muscle needs to be stimulated by a nerve, preferably. And so, when a nerve stimulates the muscle, that is the excitation phase. As the excitation phase propagates across the sarcolemma. And at this point, I encourage you guys to watch both the concepts related to membrane potential and action potential. So this will help us to interpret what's going on through the excitation-contraction coupling cycle, in which an action potential travels across the entire sarcolemma into the T tubules and stimulates the release of calcium. Once calcium is released, we're going to see what's known as the cross bridge cycle. So again, the major steps to a muscle contraction equal excitation, when the nerve stimulates the muscle. Number two is excitation-contraction coupling. That is when the action potential travels across the sarcolemma into the T tubules, into the muscle itself, and starts the release of calcium. Once calcium is released, that's the coupler. The coupler itself could be identified as calcium. And then once the calcium is released, we begin the cross-bridge cycle. Calcium binds to the regulatory proteins located on tropomyosin. Once that occurs, on troponin. Once troponin changes shape, tropomyosin reveals its bindings, opens, moves, allowing the binding sites on actin to be revealed. And then we get the cross bridge cycle, the actual cycle that it causes for a muscle contraction to occur. So, in closing here, what we're going to do is I'm going to just show you a preview of a video that's going to kind of help us demonstrate this, and I'll talk it out as we go. And what you can see here is first you have the nerve. So there's the nerve with the muscle tissue in its long cylindrical shape. Where the neuron, that motor neuron, meets the muscle, we have what's known as the neuromuscular junction. Right? And so it's the muscle fiber itself attaching to the motor neuron, which is specific to the muscle. And so the makeup of a skeletal muscle is, as I've reported, the sarcomere is the functional unit of a muscle contraction. And so a muscle fiber is made up of a number of these fibrils, and the sarcomere is made up of this alternating thick filament, thin filament. And when it contracts, what we see is what's known as the sliding filament theory. Sliding filament theory here. The two myofilaments, right? And so it's basically that we're having these two proteins contracting and sliding against each other. And so the thick filament is myosin. It is anchored in the midline. The thin filament is actin. It's anchored at the Z lines on the periphery. And so again, that shortening process is again the sliding filaments, the myofilaments as we may record them. Now, this is the stages of the cross bridge cycle. You can see that myosin is doing the pulling. It's pulling actin in towards the middle. So we're looking here at what might be referred to as the cross, and you can see referred there as the cross bridge cycle itself, where again, these myosin stays stationary, actin starts pulling towards the midline, the sarcomere itself shortens. These proteins don't necessarily uh, actually shorten themselves. And so, again, the energy that's required for a muscle contraction to occur is right here at the cross bridge cycle occurrence. So it requires ATP to actually detach. So the energy occurs in two ways. When ATP binds to the myosin, what's going to happen is you're going to get a detachment and then a repositioning of the myosin head. So there's detachment, there's repositioning. And so the actual power stroke, as it's called here, is passive. It's the energy that's required to detach the actin from myosin and then do to actually reposition it. Now, here we have the excitation contraction coupling. Remember, calcium is our coupler. So when we have calcium come and bind to the regulatory protein troponin, it's going to change shape, allowing tropomyosin to move away from the binding sites of myosin. And so now we can have a cross bridge actually occur. Remember that the power stroke is passive, doesn't require energy. Energy is used to uh, reposition. And really, calcium's housed in the sarcoplasmic reticulum. So what happens is a nerve will stimulate the sarcolemma, remember the cell membrane of the muscle, by binding to the receptors. Those receptors are going to allow sodium in, causing a depolarization and action potential to travel across not only the sarcolemma but into the cell itself, and then causing a release of calcium. Calcium binds to troponin, cross bridge cycle occurs if energy is available. it will continue to occur. So hopefully you were able to at least identify some of those steps there, or the steps of muscle contraction. I'll explain each of them in detail in the next videos, but first to get you a conception of how these proteins are going to interact with each other. Stay tuned for more.